Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for the webinar. Today, Dr. Stephen Maren will present Can Traumatic Memories Be Erased? We scheduled today's webinar well in advance of the coronavirus crisis, but the topic is especially relevant today. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $408 million to fund more than 5,900 grants around the world. I'm delighted to introduce Dr. Stephen Maren. Dr. Maren is Distinguished Professor in the Department of Psychological and Brain Sciences at Texas A&M University. He's a member of our Scientific Council and was a 2017 Distinguished Investigator Grantee. Today's webinar will begin with Dr. Maren's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now I'm pleased to introduce Dr. Maren. Steve, the floor is yours.
All right, great. So yes, once again, thanks for the opportunity to speak to you today. I know these are challenging times. Many of us are sheltering in place. I know that I've been doing so for over a month now, but this is an opportunity to talk about some interesting new science and perhaps will serve as a distraction from some of the things that are going on around us. I'd like to thank the Brain and Behavior Research Foundation. As Jeff mentioned, they awarded me a Distinguished Investigator Grant in 2017. And some of the work supported by that grant I'll talk about towards the end of the presentation. Some disclosures to start out with. So as Jeff said, I'm a faculty member at Texas A&M University. My research is funded by the National Institutes of Health. I receive an honorarium as editor-in-chief of behavioral brain research. I do not have any corporate relationships or interests. And another important point is that I am a basic scientist interested in the neurobiology of learning and memory. And I'm not a clinician. So while the work has lots of translational relevance, I believe, we're really interested in finding out how the brain comes to store emotional memories and how we might target those memories and perhaps erase them as an emerging therapy, but we're clearly far from that at this point. Okay, so the world is a dangerous place. I mean, there's been all sorts of incidents recently, and as Jeff pointed out, we're actually experiencing a fairly traumatic time right now. Trauma, fear, and terror are commonplace experiences that we all have at one point in our lives. Now, our brains are built to detect threat and to learn about threats in the environment. Forming memories of traumatic experiences is completely normal and, in fact, highly adaptive for our survival. If we can't recognize threats and avoid them in the future, we're not likely to survive and our genes won't be passed on to the next generation. But in some cases and in some individuals, these traumatic experiences can lead to disabling forms of psychopathology. And one form of psychopathology that we're particularly interested in is post-traumatic stress disorder, which is in fact sort of fundamentally a disorder of memory insofar as it's anchored to a particular experience in time. PTSD is one of many anxiety disorders, and collectively anxiety disorders are some of the most prevalent mental health disorders in the world, clearly debilitating, extreme cost to both individuals and society, and a problem that has focused many resources in the scientific and clinical communities to address. So this is some data from the National Comorbidity Survey. This is older data, but it's relatively stable over time, just to give you a sense of how prevalent anxiety disorders as a class are. So any anxiety disorder, the lifetime prevalence is around 25–30%. And there's differences, as you see here, between men and women in the prevalence of these disorders. In many cases, women are more likely to experience an anxiety disorder than men. That's particularly true of phobias, PTSD, generalized anxiety, and others. Now, we've been very focused on PTSD because it's very tractable to laboratory studies for reasons that will become clear later in the talk. Namely, it's a disorder in which an experience sort of anchors the trauma. So what is PTSD? Post-traumatic stress disorder, as I said, is anchored to a traumatic event. There's usually one signature trauma that people often refer to in the genesis of the disorder, although some individuals with PTSD have experienced repeated trauma. So while there can be a singular event that's often a causal, a history of trauma has broad implications for developing PTSD. PTSD develops in some individuals, and of course, this is an interesting aspect of the disorder that we don't fully understand. About only 10 to 12% of individuals that experience a trauma will go on to develop PTSD, but some of the precipitating events are that a natural disaster, combat trauma, terrorist attack, domestic sexual abuse, death of a loved one, motor vehicle accident, physical or sexual assault, an emergency medical crisis, and one could actually conclude COVID-19 among these sorts of things that are traumatizing people presently. So it's a direct or perceived threat to one's life that results in some individuals in the development of PTSD. What are the symptoms of PTSD? Well, one of the core symptoms of PTSD is re-experiencing the trauma. And this is, of course, relates to the fact that PTSD is anchored to a particular experience. It's anchored to a particular traumatic experience. So patients with PTSD re-experience the event in different ways. One way could be flashbacks that happen in the moment during waking life, sort of being transported to the time and place that the trauma occurred. And then, of course, sleep disturbances are common with PTSD, with many individuals experiencing nightmares or reemergence of the traumatic memory during sleep. Now, these aren't the only symptoms associated with PTSD. Other symptoms include numbing and social withdrawal. Avoidance of trauma reminders is a very common PTSD symptom. And this can be both physical avoidance of places, people, objects associated with the trauma, but it can also be sort of a cognitive avoidance where people try to put out of mind the memories associated with the event, thoughts, and other things that might remind them of the event. There's clearly physiological changes that accompany PTSD. Hypervigilance, where people are often scanning the environment, for threats, potential threats. And then, of course, changes in physiological reactivity. So there's increases in acoustic startle responses. People startle more to loud noises, changes in sensitivity to light and noise and other sensory stimuli. So there's a complex disorder and a complex pattern of symptoms associated with PTSD, but we've come to learn quite a bit how this works in the brain, and we're going to talk about some of those brain circuits as we move along. One thing that's important to appreciate about PTSD is that it often occurs along with other disorders. PTSD and substance addiction, for instance, are highly comorbid and occur together. Suicidal thoughts and depression often accompany PTSD. So when thinking about this in a human population, it's important to appreciate that there is a constellation of syndromes that are often networked with this core PTSD disorder. We're going to focus primarily on PTSD, and the animal work really kind of focuses on PTSD solely. So how do we bring this into the laboratory? So the way many investigators have been able to bring this under experimental control is to use basic forms of learning and memory like classical conditioning, or what we call here Pavlovian conditioning. So Pavlovian conditioning is a fundamental form of associative learning, and it can model certain aspects of PTSD. So in a typical Pavlovian fear conditioning procedure, which you can run in either a rat, mouse, or human, you set up a situation in which you have stimuli that the experimenter controls. For instance, in this example, an audible tone stimulus, which here is called the conditioned stimulus, which before being paired with an aversive stimulus really is fairly neutral to the subject. That is, it might produce an orienting response, but it doesn't certainly produce fear. But if you pair this neutral conditioned stimulus with a aversive unconditioned stimulus, in this case, an electric foot shock to the foot of a rat, or a wrist shock or loud noise presented to a human, pairing those two stimuli together will ultimately result in a fear response to the CS. So that's called the conditioned response. And you might remember from your intro psychology class, if you ever took one, that this is very similar to what Pavlov described in his dogs, in which he paired auditory stimuli with meat powder in the mouth and produced salivation. Well, in this case, we're presenting a tone with the foot shock, which produces a learned fear response. So as I said, this can be done in both rats and humans, and so allows for a lot of translational convergence in the laboratory, and it's under high levels of experimental control, parametric control. So experimenters can control all of the duration, timing, intensity of all of these stimuli, and it's a procedure which can model aspects of PTSD. Clearly, this is an aversive memory that's being formed here. We don't necessarily believe that rats that undergo this procedure, or humans who undergo this procedure in the laboratory, develop PTSD, but it models the acquisition of an aversive memory that's clearly core to PTSD. So using these conditioning procedures has actually given us considerable leverage into studying the phenomenon in the laboratory, not only in terms of how aversive memory or a traumatic memory might be learned, but also in terms of how we understand behavioral therapies for PTSD and other anxiety disorders. So using the learning and memory terminology, I can kind of tell you about how we think about this in the laboratory. So we believe that this fear conditioning procedure, this Pavlovian conditioning procedure, is an episode of learning in which stimuli associated with a trauma come to evoke a fear memory. And this would be similar to someone who experiences a combat trauma, for example, where there's an explosion and an injury. And so now stimuli associated with that traumatic event, like bright lights and noises, later come to evoke fear in the way of someone with a combat-related PTSD might have a fear response at a fireworks show, for example. Now, there's another form of learning which we've studied extensively called extinction, which is, as we'll see, core to therapeutic interventions for PTSD. So with extinction, cues, these trauma-associated stimuli, are experienced in the absence of the aversive event. So while these cues will initially evoke high levels of fear, if you can see from the schematic here, fear is learned during the conditioning procedure, and then during the extinction procedure, even though there's no aversive stimulus present, fear is still high. But as repeated experiences of these trauma-associated cues in the absence of the aversive stimulus will ultimately reduce the fear response, leading to a new memory, an extinction memory. Of course, many individuals that undergo therapeutic procedures for anxiety and PTSD undergo relapse, and fear and trauma-associated memories come back. And there's a number of factors which can drive the relapse of fear: time, passage of time after therapy, new stressors one might experience, or changes in context out of the therapeutic context. All of these things can cause fear to relapse. And so this tells us some important things about the nature of conditioning and extinction memories that that'll say a little bit more about in the next couple of slides. So how do you treat PTSD? So there's two approaches: cognitive behavioral therapies and/or medication. These are sometimes used alone or in combination with one another, and it's tailored to an individual's needs and how they respond to in each domain. CBT is highly effective, however, and it's generally thought to be more effective than medications for treating PTSD. One very successful form of CBT is called prolonged exposure therapy. And this is a procedure in which individuals with PTSD confront their fears in a safe setting. And this might remind you of what I said in the previous slide about extinction, which is that you're experiencing trauma-related stimuli without an aversive consequence in a safe setting. So essentially, a major component of prolonged exposure therapy is extinction learning. It's essentially an extinction procedure in part. That's not all that it is. There's more to prolonged exposure therapy than mere extinction, because patients, individuals with PTSD, are actively engaged in managing their fears. There's a supportive therapist who often uses relaxation techniques as well, which may involve different types of learning. So there's more to CBT and prolonged exposure than extinction, but extinction is clearly a core aspect of the procedure. And the goal, of course, of these therapies is to reduce fear and other PTSD symptoms. Now, as I said previously, one of the issues with these behavioral therapies is that fear can relapse under a variety of conditions. In other words, the extinction memory that's formed during the therapeutic process or during an extinction procedure in the laboratory is labile. So what that means is that while extinction can suppress the fear memory, it can result in symptomatic relief and can help individuals deal with the traumatic memory. It does not erase the traumatic memory. If it were to erase the memory, you wouldn't observe this relapse phenomenon, which is quite common. So what this tells us is that extinction is in fact not an erasure of the traumatic memory. It's not like the extinction or therapeutic process overwrites the trauma. Merely it generates a new memory, a new safety memory, which essentially competes with the fear trauma memory for expression and thoughts and behavior. This feature of extinction was actually appreciated by the Polish psychologist Konorski, who wrote in his book in 1967, in the course of extinction, the inhibitory connections are being formed side by side with the totally preserved excitatory connections. So the idea is that extinction is a new form of learning, that it ultimately inhibits the fear response, but it doesn't erase the fear response. So after an extinction procedure in the laboratory with a rat, or in a human, or in an individual with PTSD who's undergoing CBT and prolonged exposure, these procedures essentially ultimately result in the formation of a new extinction memory, which can inhibit the fear memory, but doesn't erase the fear memory. And these two memories live on in the brain, competing with one another. And as the extinction memory fades with time, or is disrupted by stressors or changes in context, the fear memory can reappear and cause relapse. So this brings us to what we know about the neurobiology underlying fear and extinction. And we've learned an amazing amount about the brain substrates of these processes. Here's a human brain. It's a section right through the middle of the brain, so you sort of see the interior of the brain. There's three brain structures which are key players in these phenomenon. The amygdala is essential for the fear conditioning. It's the place where trauma memories are formed and stored. When extinction procedures are delivered, new circuits come online that ultimately inhibit fear memories in the amygdala. One of the brain areas that's critical in the inhibition of fear is the prefrontal cortex. So this is up in the front of the brain, and it has projections to the amygdala, and this part of the brain inhibits the amygdala to suppress the expression of fear. As I said, there's two memories that are stored in the system. There's likely more than two memories, but for simplicity, we'll talk about the fear memory and the extinction memory, and they compete with one another. Well, it turns out that there's considerable work that indicates that one of the brain areas that's critical for regulating which of these memories actually is expressed is the hippocampus, which is critical for learning and memory, episodic learning, explicit memories about what, where, when, and who, pieces of information around an event. And the hippocampus is critical for regulating the expression of fear or extinction in this circuit. So we think hippocampal connections with these other brain areas is involved in fear relapse. So this begs the question, though, if extinction procedures and prolonged exposure don't ultimately remove the traumatic memory, but leave intact the fear memory, and if extinction memories are labile, this is not necessarily going to be a long-term solution to healing an individual with PTSD, for example, because extinction memories, as we know from considerable laboratory work, tend to be very labile and making the fear susceptible to relapse. So this brings us to this possibility that, well, perhaps one clinical manipulation that could prove more effective in this domain then would be to attempt to erase the traumatic memory, to remove the fear memory. And so this is going to be really the focus of the last part of the talk, is whether we can in fact erase traumatic memories, and some cautions and caveats around this issue. Now, you might have seen this movie Men in Black, several years ago, whereby memory erasure was used by the agents to suppress, erase memories of alien encounters, to try to keep those alien encounters secret. They used this neuralizer to accomplish this erasure. So very much of an idea that's been present in movies and books as science fiction. Another example is another more recent movie called Eternal Sunshine of the Spotless Mind, in which a couple who are in a soured relationship decide to have memories of one another erased, and then to see whether they could then start fresh. And again, this idea sounds very science fiction-y, and doesn't seem would be a real possibility. But as Liz Phelps and Stefan Hoffman just pointed out in a recent review published in Nature, this idea that you can edit or erase memories has really moved from the realm of science fiction to clinical practice. You see here in their abstract, they say the science of memory editing is more complicated and nuanced than fiction, but its rapid development holds promise for future applications. So what are the advances that have brought us to the cusp of using memory editing in a therapeutic context? As I say, there is a long way to go, but we're now understanding so much about the neurobiology of memory formation that we can begin to think about targeting these traumatic memories. So this is a figure from Phelps and Hoffman paper, which gives a really nice illustration of the time windows in which one might intervene to edit or erase memory. And it turns out we've known for over 100 years about this time window, which is that soon after a piece of information is learned, a memory is formed, and that would be happening at time 0 here, right in the corner, and this is time passing on the bottom. But soon after a memory is formed, we've long known that it is labile and sensitive to disruption. So there's a variety of manipulations or occurrences that can cause amnesia. So for instance, head trauma, if you have learned something in your class on campus and then get into a car accident in which you have a head trauma, you're likely to have lost the memory for that information that happened just prior to the head trauma. We've also appreciated that there's interventions that are commonly used for depression, like electroconvulsive therapy, that both in humans and animals can have effects on memories that have not yet consolidated. In the laboratory, you can administer protein synthesis inhibitors to animals, and that will also cause deficits in the consolidation of memory. So the notion is that soon after something is learned, there's a period under which it's very vulnerable to disruption, but as time passes, that memory becomes consolidated into long-term memory. And this consolidation process is extremely lengthy in humans, certainly years, and in some cases, maybe even decades that this process is happening. More recently, it's become appreciated that there's a second period of vulnerability after which a memory is reactivated. So the memory's already been consolidated into long-term memory and then is reactivated through a reminder, for example, or something else. That again brings it into a state of lability where it is again sensitive to disruption. This has led to the idea that there is a second process called reconsolidation, through which retrieved memories have to yet again be kind of repackaged, reconsolidated, and if they are not allowed to undergo this process, the memory is lost. And so there's work showing that protein synthesis inhibitors, ECT, and drugs like propranolol can disrupt the reconsolidation of memory. And I just want to point out that there's a number of individuals that have been really important in looking at this side of things: Susan Serra, Kareem Nader, Marie Monfils, and Joe Ledoux. Actually, Kareem, Marie, and were working in Joe Ledoux's lab when they made some important contributions in this domain, but really giving us a new sense of a period of time, a window in which memories can be edited, perhaps, or even erased after reactivation. So this, with this knowledge in mind, Maureen Croes in 2014 showed that in humans and individuals suffering monopolar depression who were undergoing ECT to treat their depression, they arranged a study to examine whether ECT could be effective at disrupting reconsolidation of memory. So the patients were divided into 3 groups. All 3 groups studied some emotional stories, 2 emotional stories. And then a week later, one of those stories was reactivated. There was another story that was not reactivated. And when I say reactivated, they were given reminder cues about the story, something that would bring the story back into mind. In some of these subjects, then, this was done right before their ECT procedure. Within 4 minutes of reactivating the story, they underwent ECT. And then they were tested for the retention of the story 24 hours later. Another group had the same procedure done, but they were tested for their memory of the story within a couple hours of the ECT procedure. And then there was a third group of individuals that did not undergo ECT. And then they were tested for their retention of the stories 24 hours later. And really the 2 bars here, the 2 pieces of data that are most important to appreciate is that the individuals who did not undergo ECT had the best recollection of the story. The individuals that underwent ECT after the story was reactivated had the worst recollection of the story. So this is demonstrating in people undergoing ECT that a window to edit, or in this case, perhaps erase the memory had been opened, the ECT procedure was delivered, and seems to have blunted memory for the subject material that was reactivated shortly before the procedure. Of course, ECT is a global brain-wide disruption and likely has many effects on many neural systems and may not be specific to the memories that were being reactivated and targeted. We now know considerable information about select neural circuits in the brain and ensembles of neurons, groups of neurons that are involved in memory, that it's allowed us to be much more selective in targeting these memories. And some of the superstars in this field that have been really pushing it forward are the Tanagawa group. This is Susumu Tanagawa and his student Steve Ramirez and Zhulu. Steve Ramirez is now an assistant professor in Boston. Sheena Jocelyn, Paul Franklin in Toronto, Mark Mayford and the Salk Institute have made considerable progress in selectively targeting memories in the brain. So what's an example? So this is a really exciting study that came out of the Sheena Jocelyn and Paul Franklin's lab now about 10 years ago, in which they selectively targeted fear memories in the mouse amygdala. As you recall from my previous discussion of the neuroanatomy of fear, the amygdala, which here in the mouse brain is in the medial temporal lobe down at the bottom of the brain, similar to where it is in the human brain, the mouse amygdala is where fear memories are formed and stored. So what Sheena and Paul did was use a viral strategy to introduce a virus into the amygdala neurons that allowed them to capture fear memories during learning. So the virus they used would infect some of the amygdala neurons here, illustrated as these little pyramids with hair coming out of them. These are dendrites that are coming out of these neurons. So they infected some of these neurons, and the virus that they used expressed a protein which made these neurons more likely to enter the memory when the animals underwent the fear conditioning procedure. So we can essentially call these memory neurons here in the amygdala. They also expressed another protein in these same cells, these same green neurons, that was a toxin that's not normally in the mouse brain or in a mouse brain cell. And what that allowed them to do then is, after learning had occurred, they could then inject the mouse systemically with a needle in its gut with the toxin. The toxin would enter the brain and then kill the neurons that had essentially been incorporated into the memory. So after they targeted and killed these neurons in the amygdala, they essentially delete the neurons, and what they found is that the fear memory was dramatically reduced, perhaps erased. So this was a very exciting and influential finding, which suggested that fear memory, if you target the right neurons in the brain, and only a very small number of neurons, in fact, in the mouse amygdala, under 10%, that you can selectively erase a memory. Now, of course, This is a destructive procedure. It involves actual brain damage, deleting neurons, and that would not be presumably a tractable translational clinical approach moving forward because it involves cell loss. So there's other approaches, though, that have generated promise that one can capture and manipulate memories using less invasive procedures, or at least procedures that don't involve cell death. So this is an exciting study that was published a few years ago by the Tanagawa lab, by Julu, Steve Ramirez, and others, in which they used a very sophisticated viral approach to capture, in this case, neurons in the hippocampus, which is another brain area that's critical for learning and memory, and for memory of fear, in this case, fear of places or contexts. They used this approach to capture neurons in the hippocampus. And essentially what they did was they used a virus that expressed a light-sensitive protein called channel rhodopsin only in those neurons that were active at the time of learning. So this procedure is such that you could open a window in which the neurons can be captured and then tagged, expressing this light-sensitive protein. That's these yellow neurons shown here. And then they had an optic fiber implanted into the mouse's brain that allowed them to shine yellow light, and that, to shine blue light, and that blue light would then activate these neurons that had been captured during learning. So what they did was they asked if they captured this network of cells in the hippocampus active during learning, a fear conditioning procedure, when they then turned this blue light on and reactivated those cells, would the animal show fear? And that's exactly what they showed. So here's a picture of the hippocampus of the mouse. These green glowing cells are the cells that are expressing this light sensitive protein. And here you can see when they shine light, turn the light on, the animals engage in freezing behavior. Freezing behavior is a species specific defense response that rodents show. We presume that this is reflecting an emotional state of fear. We obviously can't ask a mouse if it's afraid, but when it's showing freezing behavior, we suspect it's showing a fear-like state. And you can see that when they turn the light on, the mouse shows high levels of freezing behavior. They turn the light off, freezing goes away, and so on and so forth. So this suggests that you can capture and then reactivate a particular memory in the brain by very selectively tagging groups of neurons, in this case in the hippocampus. Now, this procedure that I just described to you involves tagging memory during learning. So there's a lot of things that are happening when the animal is undergoing a conditioning procedure. It's forming sensory representations of the stimuli that are used during conditioning, the tone, the shock, the context. It's forming associations between those stimuli, which are ultimately going to result in the long-term memory. But there's a lot going on. And of course, in thinking about how one might use this sort of procedure clinically, it's not typically the case that you have the opportunity to capture a traumatic memory as it's being formed. Rather, you're often dealing with individuals who have some past history of trauma, often decades prior, in which you'd like to be able to capture and manipulate the memory. So we were interested in whether one could capture a memory, a remote memory, during retrieval. And this is the point in time when you see patients who have had a remote trauma who come and might remember their trauma or have the memory of trauma evoked during a therapeutic session. This would be the time period where it would be advantageous to be able to capture the memory and manipulate it, maybe erase it. Of course, normal recollection is driven by retrieval cues. So if you are an individual who witnessed or experienced the Las Vegas shooting, which led to mass casualties, extreme loss of life, litter on the ground, all of these plastic cups and water bottles and so forth, litter on the ground, plastic bottles might serve as a retrieval cue for the terror associated with this event. Now, therapeutically, clinicians use these retrieval cues to reactivate traumatic memories, and they use these cues to reactivate the memory in a safe setting with the hope of reducing fear of these situations and stimuli. That's not always possible to actually put someone back into the original situation. where the trauma was experienced. So often one uses a procedure called imaginal exposure, where you ask the person to imagine or remember the past traumatic memory as a way to reactivate it. So what we were interested in, and this is work that was funded by the Brain and Behavior Research Foundation, is whether we could use essentially imaginal cues in rats to retrieve and capture memories. So how did we go about doing this? This is work that's done by Reed Ressler, who's a current graduate student in my laboratory, where we're attempting to capture a remote memory that we can later then manipulate. So the idea was, again, to capture hippocampal neurons activated by imaginal fear memory retrieval. So you might be asking yourself, how can you do imaginal fear memory retrieval in a rat? Well, it's difficult, and this is perhaps a stretch, but a strategy that we use that I'll explain to you further is we use what's called a backward conditioning procedure, in which there's an aversive event, a shock, in a particular place, which causes fear to this place, and that event's actually followed by a tone. So unlike the procedure I described to you before, where the tone precedes the shock and the animals learn that the tone predicts the shock, this is the opposite. The tone follows the foot shock. Now, why this is important, and I'll show you data that speaks to this in the next slide, is that the tone, in this case, we believe can evoke fear, and it does evoke fear, but not by virtue of its direct association with shock. It does so by reminding the animal of the place where shock occurred. So we think this is something like imaginal reactivation retrieval in humans. And that's another discussion, but that's what we think is going on. So what we did in this experiment, we used this procedure when we reminded the animal of the place that was associated with a shock. We had viruses in the hippocampus that allowed us to capture those activated neurons. All these red dots here are activated neurons in the hippocampus. And then those neurons were also expressing a receptor rather than the light sensitive channel that the Tanagawa group used. We're expressing a receptor protein here that can be activated by a designer drug called CNO. So we could basically give the animals this drug and then use that to reactivate these cells in the brain. We wanted to know was whether activating these captured neurons in the hippocampus would cause a fear response. So let me just go back and say a little bit more about backward fear conditioning. Why backward fear conditioning? So here's the procedure. You shock the animal, and then a tone comes on. Now when I say shock the animal, this is a relatively mild foot shock. It's aversive to the animals. It doesn't cause tissue injury or damage, but they don't like it. So this procedure essentially results in fear. They will fear this tone, but it's not because they've learned to associate it with the US. What we believe is happening is they learn that the place in which the shock occurs is dangerous, the context in which shock occurs is dangerous, and the backward CS retrieves that memory. So this is work done by Travis Good, who's now a postdoc at Harvard with Amar Sahay, and Reed Ressler, a current graduate student in my lab. But what they did to demonstrate this phenomenon is the animals underwent fear conditioning here. You can see during the conditioning procedure, they're showing high levels of freezing. You get 4 groups of animals. 2 of those groups underwent the backward conditioning procedure. Another 2 groups underwent the standard forward procedure where the CS happens before the US. And then half the animals in each of those groups underwent an extinction procedure. But in this case, the extinction procedure was they were extinguished to the place in which conditioning had occurred, meaning that they were just placed in the box where they had previously experienced the tone and the foot shock, but they're just placed in the box and nothing happened. So they're extinguishing their fear of this place. They're learning that the place is safe. And what's really interesting here, and I'll just point you to this bar graph here, is that that extinction procedure only affected animals in the backward conditioning, didn't do anything to animals in the forward condition. And what the extinction procedure did was it reduced fear to the CS, the backward CS. So what this suggests is that the fear to the backward CS is being mediated by the context. It's being mediated by a memory that the place is bad. And if you extinguish that place memory, then you reduce fear to the CS. So the architecture of the memory, we think, looks something like this, where the animals experience an aversive event, the US, in a particular place, and that's a strong memory that's formed. Because the CS is following the US, they don't form a strong CS-US association as they do when the CS precedes the US. So in order for the CS, the backward CS, to produce a fear response, it goes through this representation involving the context shock association. And if you extinguish this memory here, then the CS is no longer able to evoke fear. So this is why we think that this backward CS is essentially a reminder that retrieves, and we call this a covert retrieval because it's sort of an indirect retrieval of this place memory, which then allows us a window to manipulate it. So in the full experiment to examine whether we could capture and then reactivate this memory and potentially manipulate the memory, we did something like this. So animals were placed in a particular context where they were just exposed to this context. This context is going to be ultimately where we're going to do the capture manipulation. So we wanted the animals to be familiar with the context. They then underwent the backward conditioning procedure in which a US preceded a CS. Now again, this causes a fear response to the place, and the Q, the auditory CS, will also cause a fear response, but we think it's because they're remembering the place. During this phase of the experiment, we used our tools to open a window to manipulate the memory. Half the animals were presented the tone CS, the backward CS, the other half were not. And so it's in these animals we're imagining that they are remembering the place in which shock occurred, and now we're going to capture those neurons and express that receptor protein that will enable us to turn those neurons on at a later time point. So then in this later time point, they're put in a third context that they've never been in before. It's a place that they would normally not show any fear to. And then they were given this drug, CNO, to reactivate this ensemble of neurons that we had captured. And as you can see over here in this bar graph, animals that underwent the capture procedure and retrieved this memory, when we gave them the drug, they showed high levels of freezing compared to animals that had the same retrieval experience but were not given the drug. In addition, we had a control group that underwent this procedure but never heard the tone, so they never retrieved the memory of the context. And for these animals, the drug produced no fear. So this is another example of a situation where we can selectively tag a group of neurons, capture them, in this case during memory retrieval, and during, in fact, an indirect memory retrieval, something like an imaginal exposure, and then manipulate the memory. And then ultimately what we're interested in is whether we can, here we're showing that we can reactivate the memory. Ultimately, we're also interested in knowing whether we can perturb or erase the memory. As a first step towards that, we used an old pharmacological approach to determine whether we can erase this covertly retrieved memory. So what we did in this experiment, this is again work done by Travis Good and Reed Ressler in my lab, is we reactivated the contextual fear memory with a backward CS. So we present this, animals have undergone the conditioning procedure, then we do this retrieval procedure, so there's no shock happening at this point. We present the backward CS to the animal. We think it's causing them to remember the place where bad things happened. And then immediately after this experience, and again, this is a period of time where memory is labile, this presumably period under which it needs to undergo reconsolidation. We know that reconsolidation through the work of Cream-Nader and others, it's sensitive to inhibition of protein synthesis. So we injected, immediately after this retrieval experience, injected into the hippocampus a protein synthesis inhibitor called rapamycin with the thought that we might be able to impair the reconsolidation of the memory. And so this was the design of this experiment. We had animals that underwent either forward conditioning or backward conditioning. We expected that only these animals would be sensitive to the manipulation. And I'm only going to show you data from these animals in this bar graph for simplicity. The animals underwent conditioning. They then underwent this reactivation procedure where just the CS was presented, where we're thinking that they're retrieving a memory of the place where the bad shock happened. They then immediately get an infusion into their hippocampus of rapamycin. And then 48 hours after that procedure, we put them back in the place where shock happened and asked whether they have fear of the place. Animals that underwent the vehicle procedure, that is, they had no inhibition of protein synthesis, this is a control group, they showed high levels of freezing, whereas animals that had the protein synthesis inhibitor injected into the hippocampus showed a reduction in freezing, which suggests that we were able to, in part, perturb the memory, possibly erasing part of the memory. So those are the experimental data that I wanted to share with you, and we're running towards the end of the presentation, but I just wanted to raise some important cautions, caveats, and questions at this point. So one thing that's true, and we've known this historically in the literature for decades, is that manipulations that cause amnestic effects, memory impairing effects, are often the result of retrieval failures. That is, the information is still in the brain, it's still in the neurons that encode the information, but it's a failure to retrieve that information, not a loss or erasure of the information. It's as if the information is forgotten but not gone. Now, prior work that I've described to you today, including our own work, has not demonstrated that the memory loss, for instance, in that last rapamycin experiment, is complete or permanent. None of those previous studies that involved erasure or reconsolidation deficits have actually demonstrated a complete loss. That is, there's been no tests of saving. So this is an important thing to move forward experimentally, is to ask whether, in fact, erasure has occurred or whether these are temporary retrieval failures and that the information still lingers in the brain despite our attempts to delete it. The other important question, caveat, is that memories are highly associative. We know this from our own personal experience. If you smell a particular perfume, you might be reminded of an old lover or a family member or something like that. And it invokes all sorts of other memories, like maybe where you last encountered that person, a memory of a date you went on with the individual or something like that. And so in these experiments where we're trying to selectively target memories, one concern is that by virtue of having a highly associative network in our brains, that you can never really be particularly selective about the memory you target. You might inadvertently target much more than the memory you're trying to. And then another caution, and this is a major caution, is it ethical to think about erasing, editing, manipulating memory? One way to pose this question is what are we as individuals or even as a culture without memories of the past, good or bad? What if we could delete memories of terrible world events like the Holocaust or something like that? That seems to be not a good thing to do culturally. We have to preserve our histories, and so by manipulating memory, whether in individuals or otherwise, we may have implications that are much larger than the clinical context. So with that, I'd like to conclude. These are the points that I hope you derived from the presentation, which is that exposure therapies, which involve extinction, don't erase traumatic memories, and that's evidenced by relapse. Retrieved, reactivated memories open up a new window after which they can be edited, potentially erased, becoming susceptible to disruption. And modern tools now allow us to potentially selectively target and manipulate the groups of neurons representing memories, so-called engrams, allowing us to perhaps reactivate lost memories or dampen, suppress, or erase traumatic memories, possibly. As Phelps and Hoffman noted, there is a move to translate this progress to the clinic, moving the work from the bench to the bedside, but this, of course, has to proceed with many cautions and caveats in mind. Lastly, I'd just like to thank all the people in my lab. I've tried to call out the folks that were most involved in the work, but I have a robust and vibrant lab group who are all now working at home, and we miss seeing each other, I think, all the time. And so hope all those folks are doing well. And I'd like to, again, thank the sources of funding, and also note that Susumu Tanagawa and Jiraj Roy graciously donated some of the viral vectors that we used in the work. So thanks, everyone, for your attention. I'm happy to take questions in the last minute or so.
Thank you.
Any questions,
Steve.
Jeff, from the
excellent
panel?
presentation. A lot of information. You were able to present it in a way that we. Yeah. Do you hear me? Hello?
Yes, go ahead,
Do you hear
please.
me now?
Yep.
Hello?
I do.
Yes, we can hear you.
Do you hear me?
Go ahead.
I hear you, John.
Yes.
Okay, good. Okay, good. Excellent. Good. I'm going to ask one question, and you sort of alluded to it at the end, and it's something people have been asking, and it's not direct to this research. The presentation was outstanding. With the current coronavirus crisis, how is research working? What's going on in your lab? Are people able to work remotely? What's happening with any lab animals? Give us a little sense of what's going on over this period of time.
Yeah, so it's been really devastating to research. My lab has been shut down now for some period of time, and we dramatically reduced our animal colony only to essential breeding animals. Human research has been shut down now for some time for obvious reasons. And so there's a great concern that this very important work that we're doing and many other labs are doing in many other fields and disciplines is going to have a hard time to recover from this shutdown. Obviously, this is affecting everyone dramatically in all walks of life, whether it's researchers in the lab or delivery drivers, or people working in manufacturing and so forth. But to the research community, this is especially damaging. And so we're hopeful that Congress will see its way to dedicate some funds to allow research grants that are now on hold to be extended so that the important work that so many people are doing on so many problems, including on COVID, can move forward.
Thank you. Thank you for that update. Steve, I want to again say thank you for the presentation today, for the work that you're doing, which certainly is helping our understanding of these issues and ultimately will bring about new types of treatments that can help many people. I also want to thank everybody who joined us today. 100% of all donor contributions for research are invested in our grants to scientists. All of the research we fund is made possible through supporters like you.